to 9 of 4, our talk on data types.
Thank you, Justin. Thanks to all of you for being here. You've just heard a sort of whirlwind tour of the data management and sharing plan and policy, and then some of the possible ways to implement your support for it. We're now going to go back again from that overview to a deep dive into the 6 elements of the data management and sharing plan. So we're going to repeat what you've already heard, but at a much deeper level. Please feel free to put questions in the chat or comments, and I'm going to move on to element 1, data type, which sounds like it should be very straightforward, but actually has a lot of opportunities and interesting components to it that can really be used strategically in helping your PIs or researchers to write their data management and sharing plan. So, one of the things to keep in mind as people who support PIs, most of us librarians, some of us research administrators, and other folks in the Office of Research, is that PIs sometimes need to slow down because, as this comic shows, they're thinking about very complicated issues about what keeps hearts working and what keeps eyes working, and all the different complicated processes going on in the brain or the body or whatever their topic of interest is. And they don't necessarily get to the last panel where the patient says, "Can you just tell me whether I'm healthy?" And the doctor says, "Oh yeah, you're fine. All of that was just me. What's in my brain?" Because it's weird. Your body is basically made from dissolved bread. How do we even work? So a lot of times our PIs are not really in the zone for following policy. And so they need a lot of facilitation in some cases to help them keep what the policy says to keep in mind in their focus. It's not where they're mainly focused on. The policy tells us that element 1 should briefly describe the scientific data to be managed, preserved, and shared. And then it gives us a lot of different words. It's a very wall of text. That wall of text is really talking about 3 subcomponents to data type. First, what data is going to be generated or collected, or if it's purely secondary analysis, that is like a Medicaid or Medicare data set deep dive analysis, what data is going to be secondarily created. Second, out of 1 point A, the data that will be generated, which data is going to be shared? So what is all of it? And then what is some of it? And finally, in data, it also specifies what documentation and metadata will be created and shared to go with it. In a lot of cases, that metadata part is where data librarians can really help out. In addition to saying what those 3 sections are, though, the data types element also mentions some other things that we can address. So it asks, when we're talking about the type of data to collect and then share, it's looking for the type of data in a file technical sense. It's going to be Excel spreadsheets. It's going to be PNG images. It's going to be NIFTY or DICOM images. Then, and you don't need to know what those are because you're going to ask your PI when you're working with them. The amount or size estimates of those files in a broad sense. So that could be the number of files generated per participant. We're going to have 1 MRI, 3 behavioral surveys, and a bunch of biometrics. And most of that is going, except for the MRI image, is going to go into a spreadsheet. So it could be that number of files, or it could just be the scale. It's megabyte scale, it's petabyte scale. And finally, the data modality. That's a summary of like, it's imaging data, it's genomic data, it's survey data. And that's usually where the PI's mind is, not necessarily on the files that are going to be generated. But there are parts of this element to be addressed that, or that may be addressed, that can also give researchers some flexibility. So in describing the data to be collected, the data to be shared, and the metadata, descriptions may indicate the level of aggregation that will be occurring. And that means, are we going to share individual data, or are we going to share aggregated or summary data? It says in the policy that we can describe that. So in a lot of cases, aggregating may be a very useful description to include. And then also the degree of data processing that will occur. And that could be, I'm going to share raw data because I don't really have the time to get to cleaned data, or I'm going to share processed data where some parts have been removed for privacy or to protect intellectual property or because of data licenses, for example. So I think it's useful to try to use this strategically to address concerns that may arise when you're talking to your faculty and postdocs and so on, and they get concerned. This is something that we can facilitate through a data reference interview or our discussion with the faculty. How much cleaning and transformation is going to be practical by the sharing deadline of closeout of the grant? In a lot of cases, the way that research really works is there's still data transformations and cleaning and analysis happening after the grant is closed, but we have to share data by the closeout of the grant. And so it's important to take some time to talk with faculty about like what's practical in the span of time, even if it's not the final, beautiful publishable data. And then if a faculty member or other PI is concerned about sharing, maybe aggregation or summation can address some of their concerns, depending on what it is that they're worried about. But they should use it, not abuse it. Don't over aggregate because, as you heard earlier, PIs still need to share enough to validate findings. So some questions to ask when facilitating this discussion about at what level of cleaning and what level of aggregation they're going to have. Can you, the PI, if I'm facilitating for a PI, can the PI still share enough that it's more validatable and more reusable than the article methods and results sections in most manuscripts allow? I mean, a manuscript a lot of times is very short and doesn't let you give all the details of the data. So is there any way that they can aggregate but still share enough to be more validatable than what their publication might be? Is there a way to aggregate that still moves more towards openness? For example, they could add more detailed protocols as their part of their documentation, or they could commit to including unpublishable or null or not significant at p less than 0.05 or p less than 0.01 results that just aren't fun enough and cool enough to get into a journal article, but may still be what the NIH wants us to share for this increased transparency and validation level. And then finally, would the aggregated data, if they share aggregated data, would it have enough documentation that future readers can still make accurate comparisons if they try to build on this experiment or study to expand in new directions that are inspired by that researcher's work? So that extra documentation, so that those numbers that they share can be used as baselines, could be a way, if they're being very conservative with the amount of data, they could be much more generous in the documentation. A lot of times we get a little confused, and that's because it's confusing. And which data is DMSP data? Which data is covered? Again, you heard Lisa go over this. You've heard a couple of summaries now, but it's not perfectly clear what is going to be covered at any point. So here's some possibilities. If we're doing a basic science, small data sort of study, and we start with something at a wet bench, so antibodies, cells, things like that, those are definitely not DMSP data. They're not part of the policy. Even regardless of their importance for validation, anything like paper surveys, bench notebooks, biological specimens, if you can't shove it in a computer, it's not part of the data sharing plan. Once someone does a bench analysis, like maybe a Western blot, and has a gel, that gel as a physical object is usually not covered, but a team could decide voluntarily to digitize or upload if they're using a digital output blot situation. They could voluntarily address digital images and sharing digitized images. And that's something we're seeing in journal articles more, even though it's not part of the DMSP. And so if they're going to digitize them for the journal anyway, they may want to include it in their DMS plan. It's not required though, so it's in a fuzzy zone. After that comes quantification, or the point where they type the measurements into a spreadsheet. As soon as gels, microscope images, anything like that gets measured, if they're typing it into a spreadsheet, database, REDCap form, anything that's digital, that spreadsheet database collection of numbers is definitely subject to the data management and sharing plan. So as soon as they type it in, they should start thinking about that part being effective. Usually after we type in or download our spreadsheets, there's cleaning and transformations. That's things like, there's no data in this cell, it's just empty. What do I do with it? Or splitting off identifying and non-identifying variables. Anything where they're getting the data from the initial instruments or typing in or survey responses to the analysis stage. There's a long process in there before the final analysis is ready. And that whole process, including any major issues that might normally be part of their workflow, like the identification, is definitely part of the data that is in the data management and sharing plan umbrella. Then they analyze it and get out tables and graphs for publication. And at that point, it's probably not covered by the DMS policy. They could choose to include results tables and graphs for readability, but it's really the data up to just before that point that it becomes a publishable table or graph that we're talking about adding. However, although it's not data sharing, articles must be shared under the public access policy in PubMed Central. So it's just that it moves to a different policy once it becomes a publication. And if you're unsure about that, please feel free to put questions in the chat. So when talking about data to be generated, it's good to try to aim for a reference interview or a data intake interview, an answer that's technical, but not too technical. More technical than I look at it in a microscope, less technical than the gory technical details that an IT professional might have. So for example, we're aiming for spreadsheets in Excel or CSV format. We're not so much aiming for surveys that include demographics, vitals, and mental health scales, although, I mean, if they need to write that in briefly, they can, but avoiding the temptation to go into all the details about the mental health scales that really should have been in their grant proposal, but also not getting real techie with RFC compliant CSV files and the US ASCII encoded compatibility. So they're not looking for an IT report either. Definitely don't reiterate the scientific goals or methodologies. If you're doing an interview with a faculty member or talking with them or looking at their specific aims, you may need to sort of pull out just select words. And we're also aiming for estimates. We're not committing to exact counts because everyone knows that science is not exact, and you may aim for 50 mice, but end up with 53 mice because mice sometimes make mice. In that same intake discussion, as you're talking about data to be shared, it should somehow tie to the list of data to be collected, but which subgroups out of that big collection umbrella can be shared? It doesn't have to be everything. We're aiming for something that helps validation of the scientific findings that will be found. Organize it how it makes sense. It could be a very similar narrative to 1A, or you could combine them if it's a lot of different data types, a very complicated study with subawards and some text and some computer code and some surveys and this and that. If it's lots of different types of data, it may need to be a table that addresses what's collected, what's shared, what's documented, all in one summary. So it doesn't have to be 1A paragraph, 1B paragraph, 1C paragraph. If only subsets will be shared, like identifiable is collected, de-identified is shared, or individual is collected, aggregated is shared, those subsets and what defines the nature of the data that's going in there should be described in this 1B section. This is really tightly connected with element 5 down the road. It's a little bit like how you write your abstract after you've written an article, or your PIs do. Element 5 is going to be why you won't share something. Element 1B is which parts you will share after you've removed the element 5, what you can't share. So it's easiest to think about both of them at once sometimes and not just start at the beginning and go through the end. So 1B is really after the element 5 limiting considerations, what's left to share, no matter how aggregated, no matter how summary, no matter how transformed to be de-identified, if anything, because it's still valid to say, have a plan that says, my plan is that I can't share. When possible, try to move past that though. Sometimes it may be easiest to address it all in 5.1, and you could add the element 5.1 later when where you're talking about, I'm going to talk about this down here. So if my 1A says survey data and biometrics will be collected in SurveyMonkey and everything is going to be Excel files, then 1B, where I go from collection to sharing, might be spreadsheets for sharing will be de-identified by safe harbor de-identification. The spreadsheets for sharing will include de-identified responses from all respondents except those identified as having rare conditions will be shared. Those de-identified spreadsheets will be shared in Excel format plus CSV exports for future reusability. So there's a little bit of difference, a little bit more specificity, because in this imaginary scenario, I'm assuming that there's some sensitive human data that I need to specify how I'm splitting those out. Documentation and metadata for my research administration and research development friends here. Please, if you're not comfortable with documentation and metadata, consider reaching out to a librarian. We do lots of documentation and description of stuff. So this 1C, what is needed beyond the methods section to help validate and build on the data? So let's think about our example of the bear and panda. In the case of the brown bear, we got to the end with variables SAM2 and SAM3 and SAM4, and we don't know what they are. At some point in time, someone knew, though. So some things that a librarian interviewing or facilitating this writing might talk about are, is there metadata that comes out of the instruments or equipment they use automatically? Could they include that? What are their study protocols that they use in their lab for themselves or for students that rotate through? And could that be cleaned up to give a more stepwise set of definitions? We can help talk about data collection instruments or forms that they might be using. Can they PDF that so people know exactly what their participants were asked? Data dictionaries or code books of the variables, a listing of all of what is SAM2 and what was it collecting for when that downstream request comes, and other examples. You'll hear a bit more about some of these, but these are some possible ideas that you could ask about. All right, so does anyone before I hand this off to my next person, does anyone in the session have a question or want any clarification on this? I feel like this data types can be a very overlooked section because it really gives a lot of opportunity to help a PI make the choices that sometimes sometimes PIs get very stressed out about the idea of data sharing, and sometimes they really need some opportunity to make some choices in order to make themselves comfortable. Any questions or suggestions how this has worked well for you in the last R01 submission cycle, maybe? All right, seeing none, I'm going to stop sharing. Feel free to give me asynchronous, though. I'm going to pass it on to Genevieve to talk about tools, software, and code.
Hi, everybody. So yes, I'm going to talk a little bit about element 2, and we do have some good news that it is a bit shorter than element 1. They don't have the subsections. So element 2 says to indicate whether specialized tools are needed to access or manipulate shared scientific data to support replication or reuse, and names of the needed tools and software. If applicable, specify how needed tools can be accessed. So this element really asks researchers to consider important attributes such as the version of an application, as well as any needed packages or extensions needed to review and analyze the data, and really where this information can be found. So basically, this is the tools and software needed to access or manipulate the data, the specific names of the software themselves. So is it Python or is it SPSS? Is it SAS? It's basically what is the software application, how they can be accessed, so the availability of them. And this could be related to things like open source tools or purchase tools or proprietary tools. And the expected lifetime of the tool compared to the length of the data availability. So of course, if a data set, or if we know like a particular software is going to be sunsetted soon, maybe that's not the best tool to use. So just thinking about timelines in terms of software and the data itself. So this element is to place to document specialized tools such as software or equipment that another researcher may use to access or manipulate the shared data for things like replication and reuse. So I do want to, it bears mentioning that there is this element of reproducibility included in this element. And I gave some definitions earlier during the presentation. So, this element specifically mentions to support replication and reuse, and really I asked, like, what does reproducibility have to do with software? And of course, it's a lot. Reproducibility is not just about the data itself. It's about the setup and the software that was used in the original experiment. And these are really essential to have for any type of secondary analysis or reproducibility study. So it may be helpful to ask some researchers. So Nina just mentioned, this idea of a data interview or, a data reference interview while we're going through helping somebody build a DMSP. So some things to ask your researchers is can a researcher locate your data set and or your data, and or your software? And do they know what the software is supposed to do? Does this other researcher or another researcher's computer have enough information to get the software and run and produce the intended results? Do they have enough information to get the software to run and produce the intended results without contacting you, which is a big one? And are there decisions that you've made specifically about your environment or your programming that you did not document? So things that contribute to reproducible code include things like using version control, a package manager, commenting your code, documentations, readmes, things like that. So, something like R. R in different programming languages have a list of not only versions, but also dependencies and software extensions that can be used. So it's really helpful to document all of this under element 2. Code is the tip of the iceberg as it is reliant on a series of packages, native environment, and hardware. So those are some things to consider. So I do want to go over, so the NIH posted a few sample examples for writing a DMSP, and it's really helpful to look at some of these. And the first one is the NIMH, so it wasn't produced by the NIH, but the NIH is using it as an example. And this one under element 2 says all code and software that will be written to analyze the data will be deposited in GitHub for public access and will be provided as supplementary files for any type of publication. Code will be available no later than when the publication has been submitted. So there is a difference between Git and GitHub that I want to highlight. So Git is a revision or source control system that you can use locally on your computer. A lot of times it is a command line tool that you can use to stage and commit changes, and then also revert changes based on commit messages if you want. A lot of times this is used through the command line, but there are a variety of graphical user interfaces that you can use to interact with Git. A lot of times people want a public place to share code, and a lot of labs do this under GitHub. And GitHub is basically a website where you can upload copies of your Git repository. And it also has a bunch of other features related to project management, as well as taking the input from others through issues or pull requests, what have you. So a lot of times Git and GitHub are used interchangeably. And we saw some of the examples from the NIH include Git version control as well as GitHub. So I wanted to highlight that. Also, Git and Zenodo do have a link. So if you only have any type of publication, you can submit your GitHub link to Zenodo. It creates a copy of it, and that is what you can use to cite your code. So you're citing a specific version and sort of the time and place of your publication, which is really helpful. Something to consider is that code is a valid scholarly output, and you can actually add a license to it. So I do want to highlight that. So when publishing your code, it's really helpful to apply a license, and there are a lot of licenses that you can choose from. They can be as permissive as you like. A license is a document that can act as your official permissions for others to do, basically, if they want to remix it, what have you, which is really helpful. And if you don't have one, it's kind of assumed that you own the copyright, and it's a bit ambiguous on what others can do about it. So releasing your materials without a license creates ambiguity. So it's really helpful to choose a license. And there's a choosealicense.com, which is really helpful to get an overview of the available licenses that you have for code, and then choose one that is appropriate to your research. So another example that I want to mention is the NIMH sample example for clinical and MRIs. So I'm just going to read the first part of this. So it says the clinical data will be analyzed with custom Python code written using the stats model, NumPy, and pandas packages, all of which are freely available. So these are Python libraries that you can add to your Python code, as well as a 1HF MRS spectra. Will be analyzed with LC Model 6.3 software using LCM GUI, which, I don't know exactly what all that means, but I know that it is somewhat specific in saying that we're using, these particular Python packages with this other type of software and where I can find it. So they provide a URL here, which actually, I believe the most up-to-date guidance from the NIH is to not use hyperlinks, but just including the title here. But there's some other things that I want to highlight. They say that they're going to use MATLAB as well as the SPM8 toolbox. And then all of the code will be shared on our lab's website. And they will include a README file for the project as well as instructions and parameters if you choose to use the GUI-based analysis system. So there's another one as well that's related to genomics and human data. So they're using the genome analysis toolkit here, and then they're going to apply best practices workflows for a single nucleotide variant and indel discovery from a whole genome sequencing alignment files. So they're going to use SAM or BAM files here. And so these steps should ensure that the final associated results are representative of a true genotype rather than miscalls or confounded genotypes that are unlikely to replicate in independent populations. And this is actually one of the newer samples that the NIH put up, and I thought it was really, really helpful, especially if you are doing any type of REDCap support at your library. So this example comes from a human clinical and genomic data example from the NICH D. So it says clinical and laboratory data will be collected in the electronic data capture system, so REDCap, and analyzed using the open source statistical package R. For genomic data analysis, containerized open source workflows. So they're using some type of container. So this could be a Docker file that kind of encapsulate a workflow or data analysis workflow with all its dependencies already included. That are functionally equivalent to a GATTK pipeline will be used to call variants in this project and are or will be available through the NICDH's GitHub repository. So we're seeing another use of Git as a platform for sharing code. So something that I want to mention is actually software as a research output. So this is something kind of dear to me, is this idea that software and code that is being used in research does have value in terms of a scholarly output. And this is, this is something that is citable. So you can cite the software that was used in your research, which means that you can get credit for it and do in interesting ways, including all software that was used to generate and analyze data. So from Force 11 says that software should be cited on the same basis as any other research product, such as a paper or book. That is, authors should cite the appropriate set of software products just as they cite the appropriate set of papers. So if you're interested in this, so Force 11, I mentioned Force 11, this is a great resource for some of the working groups that they have. There's also Cite As, which can tell you how to cite a particular piece of software. And there's also a citation file format called a CFF, which you can add to a repository. And so you can add this to a GitHub repository, and it specifies who should get credit and how your software repository should be cited in a paper. And there's also the software citation principles as well. And something that I've also seen that I don't have it linked here, but it's also applying the FAIR principles to software, which I think is really helpful in thinking about software in terms of the FAIR principles, but also access and reuse. Oh, it's quick. Now we're off to element 6, and I can check the chat and see if there are any questions.
There was a question about citing AI that might change over time.
Oh, that is a very good question. So let me read the question, actually.
So I've already responded to the question a bit.
Okay.
So AI does present a huge reproducibility issue, sort of broadly speaking. So I've linked to a Nature article that discusses this issue that they put up in January. So So I don't have an answer for it, except to say that this is an area of real concern when it comes to the use of AI, particularly in healthcare settings where reproducibility may have, real bodily impact. Yeah, so I don't have a better answer, but I do have a lot of links to articles that continue to talk about this issue.
Thank you so much, Nicole. Yeah, I went to a talk yesterday on ChatGPT or GP3, and ChatGPT3 that talked about using that language model as someone had listed it as a co-author, and they're about the backlash with that. So hopefully we won't see AI listed as a co-author, but yeah, it is something that we need to consider. And I don't necessarily know how one would cite it, but yeah. All right,
I will at this point take over. Genevieve, are my slides visible?
I see them. They look great.
Beautiful. Okay, if anybody else wants to talk to me about reproducibility and AI, just know that I love that conversation and I'd be happy to talk to you more about it. So let's get into standards. This will be fun. So I'm going to start off with an activity, actually, just a quick short one, no breakout groups, but just in the chat, if you could write what the effective date for the new policy was. This is not really a quiz on the effective date, it's to demonstrate something. So it is January 25th, 2023, but write it however you would. Okay. I already have enough answers to demonstrate my point here. So let's get to it. If you guys look in the chat, we have a couple of responses and the same information. Barry, you've made me laugh. Congratulations. You're the first one to do that. If you look in the chat, there's a lot of different ways to write January 25th. I've put some of them down here. Barry has responded in a very creative way. I'd suggest you taking a look at the chat. Maybe it'll make you laugh too. There's a lot of ways, though, to write the same concept being the date, January 25th, 2023. But you could see, depending on where you are geographically, if you're in the States, you're going to put month first. If you are anywhere else, you'll put day first. There's a lot of different ways to write the date. And there is a standard for date writing. So the ISO, which is an international standards organization, has built a standard for date writing. It's ISO 8601, and that is year-month with 2 digits and day with 2 digits. And the reason why this is important, and you can think about this, if we had a bunch of different data sets or a bunch of different people collecting data, and they wrote down the date in all of these different ways, you'd have a real data cleaning problem. It would become a real struggle in the aggregation process. So standards help us sort of avoid those types of problems. Let's dive into it a bit more. So what is a standard? A standard is an established, generally community agreed upon way to collect, organize, or document data. So when I say community agreed upon, let's dig into that for a second. That could mean it is an established organization. So that would be like the International Standards Organization, but sometimes it could be a bit more informal than that. It might be created by a group of people through a professional or academic association. And sometimes the government participates in standard creation. The idea, though, is that a group of people have come together and they've agreed on a way to collect, organize, or document data. Now, standards help with data interoperability and our ability to aggregate data across many studies. So as with our example with date, if you are noting the same concept in different ways, it becomes really difficult to aggregate that information. However, if we note the same concept in the same way, if we use that standard for how we write down date, aggregation and interoperability become much more achievable. If you're interested in learning more about standards, you can go to fairsharing.org, which has a whole long list of the many standards that exist. So we have some caveats about standards. First, not all fields or subjects have standards, and the NIH policy does recognize that fact. Many fields or concepts within fields have multiple and competing standards. So that's definitely a challenge to figure out how to deal with it. And there are new standards being created all of the time. And sometimes those standards are incredibly specific to a field, specific to a subfield, specific to a specific action within a subfield. So the bottom line here is that it is unlikely that you as a librarian or some other support staff will be providing guidance on which standards to use for data collection specific to a discipline. It is unlikely, for example, that you're going to learn all of the standards related to microscopy image data on top of the genomic standards, on top of various public health standards. However, it's helpful to understand the concept of standards and to be prepared to discuss standards for metadata and data description specifically, since that's where many of us as librarians are quite comfortable. So let's get into the actual field description. What the field description says is state what common data standards will be applied to the scientific data and associated metadata to enable interoperability of data sets and resources, and provide the name or names of the standards that will be applied, and describe how these data standards will be applied to the scientific data generated by the research proposed in the project. If applicable, indicate that no consensus standards exist. So again, we're seeing here a recognition from the NIH that there may not be a consensus standard for a specific field. We also see a recognition that there may be multiple standards. Sometimes it's for the metadata associated with the data, sometimes it's for the data itself, sometimes it's for collection, et cetera, et cetera. So let's talk about a sample section on standards. This was taken from a sample DMP that was submitted as part of the DataWorks Data Management and Sharing Plan Challenge. And I'm going to read this out loud to you all. The images will be shared in native subject space and standard space, Montreal Neurological Institute in NIFBT compressed format. I don't know if that's the correct way to pronounce that format, which is additional support of the idea that you won't be giving specific standards advice. The data format and organization follow the brain imaging data structure BIDS guidelines, which will enable a navigation and sharing. Additionally, this format is compatible for conversion to newly developed semantic standards. This allows generating computable data objects readily used by the AI community and facilitate non-expert data analysis. The CDOs also allow integration with open science efforts, analytical pipelines, APIs, and modules for quality control, harmonization, indexing, and management engines. Now that's a lot, particularly for those of us who aren't neurology researchers. So I'm going to just pull out a few important points here. They tell us what the data format standard is, and that's the brain imaging data structure. And then they give us some information about why this is important. They talk a lot about interoperability. So that's things like those APIs and analytical pipelines. That's when we're pulling a lot of data sets together using those tools. So ensuring that aggregation is possible happens through interoperability. And we talk a little bit about what they use those standards for. So moving forward, we have 4 different examples of standards that I'm going to go through. Again, this is not going to cover all standards because there are so many, but we're going to just give some examples. We'll start with metadata standards, then go into common data elements, then data collection standards, and finally, the role of standards in repositories. Metadata standards. So many of us are probably familiar with metadata standards. Those are data standards. So if you've worked with metadata standards before, you can already consider yourself somebody who has a foot in the pool of data standards. So our example here is the data site schema. Now it standardizes how data is described with metadata, and it allows for record interoperability. So DataCite has a service called DataCite Commons, where they make metadata records discoverable across many description providers. And this is possible because all of those providers use DataCite. And again, we're talking here about aggregation and interoperability. It also allows DataCite to make records available via the DataCite REST API. So for those of you who might not know what an API is, it allows people like programmers to systematically browse, query, and retrieve metadata records. It's something that you can use if you're a little bit more programming literate. I have not used an API, just as a full confession. But it is because we have these standards set, we have these agreements about how our descriptions will be written and what information we'll put in them. That's what allows all of this technology to work. So here's the background of the data site metadata schema. And you can see here we get information like you are asked to include an identifier and that they request that all of these be DOIs. If it is not a DOI, they ask you to include the type of identifier. So this is where you would say a DOI, or perhaps you would use one of the accession numbers like they have in some of the genomic data repositories. They also request that you write in who your creator is, their full name, and the type of creator. So thinking about data sets, you may have an individual person as the creator of the data set, a researcher, but sometimes you'll have organizational creators of data sets. So here they'll have you in the schema note that the creator is an organization instead of a person. So we're going to get to our next example, which is common data elements. Now, a common data element, these are precisely defined questions paired with a set of allowable responses. So you could think of this as, we just talked about the data site metadata schema. This would be just one element of that schema, and then all of the allowable responses for that single element. So it's a smaller piece, but it still constitutes a standard. CDEs are used across different sites, studies, and trials to ensure consistent data collection and the ability to aggregate across sites, studies, and trials. I think you all might be picking up here that the key point of data standards is really aggregation. CDEs are a standard for a field. When generally we talk about standards, it is a standard for a series of fields. Okay, let me explain that. So CDEs tell us how to collect a single piece of information. Like if you're collecting age in an age group, it would be the question and then that series of allowable responses. But sometimes standards, as we think of them, are a whole series of fields. So again, like that data site example. Now I've included links here to the CDE repository from the NIH, which you can use to get a good idea of what they have available there. And they also have a tutorial on using that repository. So I'm going to go into a little bit more of how a CDE works. So I keep saying these are about single fields as opposed to a whole series of fields. Here's an example of a CDE. So the question, that defined question is, I had trouble finishing things because I was too tired. And the allowable answers are never, almost never, sometimes, often, and almost always. And you can see here the link to the CDE repository. Now, if we take a bunch of CDEs and combine them together, then we'll get that data collection form, that survey, for example, that collects a whole series of fields, a whole bunch of information from each of our subjects. So here's a field-specific example of a standard. Now, this is called Neurodata Without Borders, and you can start to see how complicated some standards can be because they standardize how you take in a whole bunch of information. So things from data acquisition, so they standardize how you note device setting, your filtering parameters, your sampling rate, your recording and imaging area. But they also standardize how you take notes on your experimental subjects. What's the species, what's the genotype, what's the age, what's the weight? Each of these pieces of information are subject to a standard. Now, one of the things that's interesting about Neurodata Without Borders is that there are repositories, notably this repository called DANDY, which stands for Distributed Archives for Neurophysiology Data Integration. And if you're going to be putting your data into DANDY, you have to use Neurodata Without Borders. Clearly, this can be a challenge because Neurodata Without Borders is a very complex standard. But what that also means is all the data in DANDY is highly interoperable, and it's very easy to aggregate across data sets from this repository. So like DANDY, there are some repositories that require use of a specific data standard for data submission. So here's another example of one of those types of repositories. The National Institutes of Mental Health have a data archive. And all data that is submitted to there must have a corresponding data structure in the NDA data dictionary. So in this case, you could understand a data structure as a standardized data collection form. So this is an example of a data structure in the NDA data dictionary. What we're seeing here is this is a question about how you note how much pain people experience while performing specific tasks. And you can see that they're collecting a lot of different pieces of information, and each of these pieces of information has a description, has an element name, etc. Oh, and I've pulled this out here for you in case you have a hard time reading small text like I do. And so what we see here is an example data element. And what this is pain threshold. It gives us a value range, so 0 to 180. What that value corresponds to is the time it takes for a participant to first feel pain when their hand is immersed in cold water. And so you could see how this is standardized from top to bottom. If we did not standardize it, people might measure time in different ways. We can see that this is measured in seconds, not in minutes. So this is how this is all standardized. So I have a few takeaways from you, which is that standards are important for data sharing, but they're very complicated and they're very messy. It is very unlikely that you will advise on what data collection standards to use, particularly for things like neurodata without borders. Some of us may be neuroscientists, but I don't think most of us are. However, you may be asked to figure out if a repository requires use of a certain data collection or description standard. And I think that's something that is much more approachable than trying to, I don't know, learn all of the standards for all the different fields of research, which I think would perhaps be impossible. And it is also possible that there will be no consensus on standards in a field. In that case, researchers are encouraged to explain that fact in their data management and sharing plans.
Thanks for watching. This video was produced by the network of the National Library of Medicine. Select the circular channel icon to subscribe to our channel, or select a video thumbnail to watch another video from the channel.